know there's 4. The concept is the same.
4 per hemoglobin?
4 for hemoglobin, there's 4 hemoglobins per unit of binding. So yeah, I mean, it comes out as 4s. Right?
Note that the dissociation curve is sigmoidal, that is, S-shaped. Because of this sigmoidal relationship, hemoglobin responds quickly and effectively to small changes in oxygen demand. If this cooperative binding did not occur, the 4 hemoglobin subunits would load or unload oxygen all at once, and much less oxygen would be delivered where and when the tissues need it.
So remember the purpose of that curve was to tell the blood how much oxygen to give to whom. So if I didn't have that curve, you just dump it all in the first organ you saw, and then everybody else suffers. Right? So your toes would always be blue. So let's review that curve.
Blood leaving the lungs has a PO2 of 100 millimeters mercury, but body tissues have PO2 levels of about 40 millimeters mercury at rest. This difference drives the unloading of oxygen from the hemoglobin to the tissues by diffusion. When blood encounters tissues at rest, hemoglobin unloads only about 30% of the oxygen it carries to the tissues. If tissue activity increases, such as during exercise, the PO2 levels of the tissues drop, and hemoglobin releases about 60% of its oxygen to the tissues.
So that's supply and demand idea. So the hungrier the tissues get, the more the blood's going to unload, and the diffusion tells it where to go. Make sense?
What happens to the carbon dioxide that leaves the tissues? Most of it diffuses into the bloodstream, specifically into red blood cells. There, much of the carbon dioxide is rapidly converted to carbonic acid by the enzyme carbonic anhydrase. The carbonic acid then dissociates, releasing a bicarbonate ion and a hydrogen ion, that is, a proton. As a result, the blood pH drops. Changes in pH...
Let's review. Blood pH drop means what in English?
Acid.
Being more acidic, right? As pH goes down, acidity goes up. So you're making the body more acidic as you have more CO2. Yes?
So this is all happening in the red, within the red blood cell?
Or the plasma.
Or the plasma, okay.
Right, so formally, 26% of the acids formed by the red blood cell, 75% is found in the plasma.
Okay.
So, but basically in the blood, it's fine.
Okay.
Either way, it's fine. But that's a different site than the hemoglobin would be. So it doesn't dark the core.
affect the shape of hemoglobin, and thus its ability to bind oxygen. For a given PO2 value, hemoglobin can bind less oxygen at lower blood pH, and more oxygen at higher blood pH. This effect is known as the Bohr shift. The drop in blood pH leads to the unloading of oxygen by hemoglobin. The protons released by the dissociation of carbonic acid bind to deoxygenated hemoglobin. Hemoglobin thereby acts as a buffer, preventing large swings in blood pH. Thus,
So let's go back to that little picture there. Not the Bohr shift, that's different. Right? So as I get more acidic, my graph shifts right or down, depending on how you want to argue the point. That's going to change the affinity of the hemoglobin. So it'll let go of more oxygen more often for the same tissues. Make sense? Hopefully this clarifies that damn curve. Cool? Okay, now it's time for your quiz.